the genome, from the bacteria to the yeast to the humans, all the regions of the genomes can be transcribed. I'm very impressed with all of the works that Tom published. And today's talk, Tom will focus on the non-coding RNA. Let's give a warm welcome.
Good morning, everyone, and thank you for coming out on this warm day. I wanted to relate to you that I have been to China several times, but the one thing about China that is really unique, and I've been to many places in the world, is that I've never come here and not been astounded or surprised by something in the culture, the people, or something. It's truly a magnificent and very rich place. So thank you for this honor and invitation. So the title today is a prelude, is a sort of start to a conversation. I hope it's a conversation rather than just a lecture, because I could stand up here and talk for a long time, but at the end of the day, I'm sure you have questions and you have ideas, and we should share with that during the course of this presentation. So I hope people will do that. Let me start. So these are sort of the guidelines or suggestions for the presentation. Please ask questions during the presentation. Don't keep them in the back of your mind, hopefully to remember at some break point to make a discussion. So please stand up or raise your hand, and I guess there are microphones that will go around, and you can make your point. You'll see a variety of citations in the presentations, and they will be very simplified. They will have the first author of the paper and the PubMed ID. And so if any of these are of interest to you, you can look it up that way. And finally, if you have any suggestions, any criticisms, any ideas that occur to you later after this presentation is over, please feel free to send me an email, right, or contact me in some fashion, and I'd be glad to talk to you about it. And so this is the beginning, I hope, of a conversation that could go on for some period of time. Okay, so the hypothesis, and the title sort of starts that hypothesis, is that that the role of RNA in the genome is really one of a facilitator. I'm not quite sure in China whether there's this culture or understanding about the concept of middle management. In the United States, middle management is a very powerful and very facilitating operation for almost any company or any organization that's large enough to need people to translate and to enact ideas that are come from the top down, or information that comes from the bottom up. This middle management makes large numbers of decisions, all right, that if they're not recognized, never get to either end of the decision tree. So my hypothesis for this presentation is that RNA plays the role of middle management, and it acts to translate and act as a conduit of the directions derived from the leadership, namely the DNA of the genome. Right? And likewise, it provides that information to elements in the cell that are also going to carry out these functions. All right? The proteins, lipids, the other RNA molecules, they bring that information to them, and then they basically get feedback from that, which ultimately goes back to the genome. So my challenge, what I'm positing here this morning, is to think about RNA in a very different fashion. It's not a subservient role. That is to say, all information resides encoded in the genome, and RNA just carries it out. It's making decisions. That RNA is making decisions by its interactions with the targets of where that information is to go. all right? And we'll talk in some detail about what that means in an actual sense. But I just wanted this general concept for you to think about as we go through this presentation, that RNA is a facilitator, all right, and a decision-making element in the cell that carries information down into the actors of the cell and information up back to the genome, all right? So it's a very powerful organization of information. So this cartoon is basically the idea that we have this one guy here who's a middle manager here, and all of these actors basically are dependent on him showing the way in which to go, all right? All right, so let's think about, let's start. This idea of RNA being more than a subservient role is, I'm sure it doesn't come as a surprise to most of you, but this is a relatively new idea. The idea that RNA is basically a product made from DNA, and its goal is basically primarily to make protein. That was the particular principal thought of actually what RNA was meant to do. And there were RNAs known for a long time that didn't encode proteins, all the ribosomal RNAs, for example. But they were thought to be a very specialized class of RNA, that the majority of the functions of RNA were in fact embodied in this translation into protein. So this is not a new concept. I mean, it goes back to Jacob and Monod, and it goes back to Watson and Crick, the central dogma where everything starts at the DNA. RNA basically just carries out this information, and that's the end of its role. All right. This perspective has a couple of issues with it. All right, and namely that as we have become much more capable of understanding the instructional content of the DNA molecule in many genomes, the one consistent idea that has sort of now permeated the literature and everyone who's interested in this topic is that irrespective of the irrespective of the organism that you're speaking about, or the genome from which this information is present, is basically that the total number of protein coding genes, all right, is relatively constant. It's relatively similar, right? And this means that if the diversity that we see in these different species resides anywhere, all right, it would have to reside either within a variation of these 20,000 genes or something else outside of these 20,000 genes. So there was this intermediate stage from the time molecular biology began to investigate the role of RNA that suggested that once we understood that RNA from a particular gene had many different variants, namely isoforms, splice variants, then the idea was that the differences in species, right, really resided in the ability to have very different isoforms in a particular cell function and in other cells not function. So that had been the standard sort of solution to this problem of having relatively constant amounts, irrespective of the genome size, of these protein coding RNAs. It's in the splicing. Splicing was everything. So that the isoforms gave us the variety we see in different cells and making up all these organisms. But the idea that there could be important functions outside of the coding regions, right, was began to emerge when we began to see this idea of RNAs being transcribed in places no one ever thought had this kind of information or had this kind of function. And what we saw was that if you plot the amount of non-coding information, if you plot the amount of non-coding space in different genomes, if you start with the single-cell bacteria, work through single-cell eukaryotic organisms, and then you get into higher eukaryotic organisms, and you begin to see this explosion of sequences that are not attributable to the coding of protein information within them. What's more, what's even more striking is that this information resides outside of the annotated genes. But in fact, when you look inside an annotated gene, you see another level of transcription embedded within those genes that are not protein coded. So this is pervasive transcription. This is RNA that is being made, as we know today, about in 80% of the entire genome. But it's not just as if it were 80% of 3 billion nucleotides. Right? It's 80% of 12 billion nucleotides, because you have 2 alleles and you have 2 strands, each of which can give rise to a different transcript with different information in it. So embedded in this genome is an incredibly large amount of opportunity to make RNA molecules, and at the bottom line is the cell takes advantage of that. It takes advantage of this space that it has, and this has been a relatively recent strategy. This has been a relatively recent strategy because you don't see this until you get to higher eukaryotes, that you have this increasing amount of space deriving increasing amounts of RNA. Now, having said that, if you go back even into single cell and the worm, for example, there is still this quite pervasive transcription going on. But because the genome doesn't have much non-coding space, that information is overlapping with the coding information that's there. And this is a very old evolutionary strategy. If you go back to viruses, all right, and arguably one of the first organized living life forms, if you want to call them that, they had very small genomes. Viruses have very small genomes, but they pack into that small genome a lot of information and a lot of different kinds of transcripts because they bury those transcripts one on top of each other in the same space. Right? So these larger organisms that went from thousands of nucleotides to billions of nucleotides, never gave up on that strategy. That strategy is still in force in the eukaryotic world, where information is being buried on top of each other, in addition to taking advantage of the very large amount of extra DNA that's present. So now we have this situation in which we have large amounts of RNA, we have very considerable organizational challenges, because if you have, let's say, 3 different kinds of RNAs, all being derived from the same region, and those RNAs potentially can have very different functions, right? Then how does the cell know which ones to make at certain amounts in certain cell types and not in others? So it brings with it this very significant regulatory issue by virtue of the fact that you have this information buried one on top of each other and also present outside of the protein coding regions. So is that clear? Does that make sense? Right? Yeah.
That means that you think that a lot of this no-coding DNA, so the single cell organism and the multi-cell organism, it seems that single cell organisms have less no-coding DNA, but the multi-cell organisms have much more no-coding DNA. And another question is that varies, varies a lot of various. Because yesterday you have found a lot of this replicated sequence in the genome is the mouse and the human difference is because this replicated sequence. And the replicated sequence most from some virus something. And this replicated sequence, it seems that most of related to this metabolism and the immune system. That means metabolism and immune system may be the interaction with the environment to talk that. That means maybe the virus drive the evolution of the organism,
Right.
And this will be very important, although they are very less.
Right. I think that's you make a very good point. So many of you in this room, perhaps all of you, realize that almost 50% of this sequence that is rich in the eukaryotic genome, the sequences that we see here in this large amount of genomic space that doesn't contain protein coding regions, that aren't 50% of that is repeat regions or repeat sequences, all right? And therefore, up until very recently, and even now, there is quite a bit of controversy, as many of you are aware, that the role of these repeat sequences, for the most part, were to be vestigial elements, that is to say, non-active elements in the genome. But we now understand that there are interesting properties of these repeat elements, which nature and evolution has taken advantage of. For example, in the retroviruses that are in the genome, right, and buried in the genome in many different copies, bring with them regulatory elements, promoters, right, which can be turned on. And guess what? Those RNAs not only make RNAs that go through the viral sequences, but also extend past the repeat regions into the coding regions. So they act in some instances like an alternative promoter for the downstream coding sequences. That's one function for these repeat sequences that have now been observed. In addition, the repeat sequences themselves, the sequences that come from them, are also provide an opportunity to act as regulatory elements for things in the non-repeat portions of the genome. So thank you. That's an important point to consider. By the way, this figure is from John Maddox's review, which is probably one of the most insightful figures that came into the public literature back in 2004. So if you've not read this review, I would strongly recommend that you take a look at it. So this sort of background, this sort of general architecture that we just discussed, is a kind of heralding of what's going to go on. How does it actually carry out? What does the organization, what is our current understanding about the organization of the genome, instead of talking about it in a general sense? So if we realize that despite the fact that we have sequenced many genomes, right, that we have the complete sequence, a working draft sequence for many, many genomes, we still don't understand what the entire information content in those genomes are. Where are all the functional regions in 12 billion nucleotides? All right? And where are the elements in that genome that control their expression, that control their processing, that control their abilities to be localized within the cell? Where are those elements? And that is basically the goal of the ENCODE project as it was conceived more than 10 years ago. Currently, as we sit here today, based on version 25 of GenCode, right? And GenCode is a group of scientists that work in collaboration with the ENCODE scientists to basically manually annotate the human and mouse genomes. So information that's coming out of the ENCODE project, right, is going directly into this GenCode project to provide a more clear and precise definition of where all the protein coding and non-protein coding genes are located. As of today, there is about 58,000 genic regions, right? Now, the 58,000 regions that we know have at least one promoter and at least one transcript being emanated from it. Of those 58,000 regions, about 23,000 produce non-coding transcripts, exclusively non-coding transcripts. We're not talking about transcripts that are buried inside a coding transcript. We're talking about exclusively outside of the protein coding regions. And there's about another 20,000 coding transcripts. Now, for those of you who have been following this for a while, this is kind of an interesting number, because this number is not increased over the last 10 or 15 years. This number has gone down. down from about 30,000 now to almost 20,000. All right, and it continues to go down because what we have mostly discovered is that most protein coding transcripts, all right, can make transcripts that connect what used to be thought of as two independent coding genes. In fact, there are many examples where two genes were thought to be differentially expressed and making independent RNAs. In fact, make the same RNA that is the same connected RNA. So the reduction, in fact, comes from better annotations to show the interconnectivity of adjacent genes, which have escaped our attention. And finally, this group of genes, well known to some of you, called pseudogenes, which were thought to be sort of vestigial genes that had, either by virtue of the RNA they made being retrotransposed on back into the genome, or a copy of the DNA made and put in some other place in the genome. These kinds of DNA and RNA versions of the gene constitute about 15,000 genetic copies. These are another form of non-coding RNAs because, as best we know, 25% of those 15,000 genes are expressed. All right? They make their own RNA. The RNA has a unique property, as many of you will realize. That unique property is that it looks like another gene, because that's the parent of that gene, right, exists somewhere else in the genome. So in 25% of the cases, there is the original coding gene making an RNA, and potentially a copy, one or more copies of that gene, right, making versions that are not coding. They are abrogated forms of it, but those RNAs are also being made. Now, if you think about it, if the RNA that is being made from the parent gene and the pseudogene copies now are vying for the same materials, like transport vehicles, like modifications, like proteins that interact with the structures of those RNAs. All of those are now present in the cell at the same time the protein coding gene is. So there is another level of regulation that clearly is going on, that the cell is able to handle these very closely related RNA molecules in the cell. So overall, there's about 34% of all genes is protein coding, and about 39% are non-coding. That leads to about 10 copies per transcribed region. So on the median number of transcripts that you see in any coding region, right, is about 10 copies, 10 different transcripts, right, being made in that locus. Again, underscoring the idea that there is a large amount of information being used in the same loci. Let me say it one more way that's perhaps even more striking, at least to my mind, is that the RNA molecule that you see, that for example may be turned into protein, and another RNA molecule, which is actually a non-coding version of it, right, has a large percent of their sequences are shared. They are shared. It's the exact same sequence. And therefore, one has a different function than the other, have to deal with the fact that you have shared sequences that have to be sorted out by the cell in its overall functions. Yes?
There are too many database and transcripts database like GenPro, or CSC.
Yeah.
But why don't we just have one, database? Because every group has his a specific definition or calling pipeline to get the annotation of the transcripts.
Yeah.
So what's the difference? Because you define some in your G code, but maybe UCSC has some other new annotation. Example has some new annotation. They are overlap maybe 80% or maybe. So another one have the link database. Another one have the circular RNA database.
Right.
how could we just get one? It's like the reference RNA database.
Yeah, that is a very legitimate point. I don't think there's a single answer to your question, but let me give you the history of this. Most of these, like RevSeq or GenCode, these databases were constituted based on a specific set of criterion, that is to say, thresholds for them to decide whether something was a real transcript and whether that transcript was coding or not. So some have very liberal kinds of criteria to bring that RNA into a database. And that's useful because if you make those criterion much more stringent, then you clearly are leaving out things that don't necessarily obey the rules of what we think we understand, for example, a coding transcript looks like, right? Or that this is a pseudogene and not another gene that's related, an ortholog of some kind. So the answer to your question is that these databases emerged when there was no real consistency, no real acceptance of a general set of rules of how to classify or identify RNA transcripts. But at that time, because there were no rules, as we sit here today, the remaining databases really reflect the thresholds and the rules that are being used to classify RNAs. One of the things, I can give you a concrete example. GenCode has very, very few single exon RNAs, virtually none. All right? Whereas other, like RevSeq, have many single exon transcripts. And the reason for that is GenCode made a decision years ago that they wouldn't use this single exon for fear that what was being analyzed was a piece of DNA, because there were no splice sections in that transcript that could clearly have to come from RNA. Right? So it's reasons like this that different databases are maintained, because the rules that they apply, all right, are not necessarily the same, and the thresholds they apply are not necessarily the same.
And I assume you put all the robust or the stringent, transcripts in your database. Maybe the other, like all transcripts, but not that robust, will be another field or like golden line for the future study. So should we have you put all the robust in this annotation, but we put maybe minor transcripts in another database, then we can for researchers to search.
Yeah, my suspicion is that there's always the concern about false positives, right, in a database. And GenCode is very conservative about what goes into the that database. By necessity, then, they're missing things that are likely to be important. All right? But the one thing you get from this kind of approach is that you have a consistent, principled way in which you can define that this transcript is either coding or non-coding, and whether it's a real transcript or not. But as I indicated, the single exon transcripts are almost totally unrepresented in the GenCode database, but they're present in other databases. So clearly, if you think you're working with a single exon transcript, GenCode is not the place to go. Okay. Anything else? Anybody else? Okay. So this ability to get annotations, the number of annotations to be increased over time, has led to a variety of consequences. And that is, for example, that if you now look at the comparison of 3 species, which we did as part of the model genome project for ENCODE, is that you see that the average length, the gene span for humans, is now up to about 4.25, 4.5 in terms of its median length. Whereas the gene lengths of the mouse and the worm the fly and the worm are much smaller, even though these organisms have genes that are many times the size of what we see in human, but they fall at the sort of the shoulders of this distribution. The off regions are fairly similar. The CDS exon lengths are fairly similar, and what is the number of CDS exons per gene for human is far exceeds what we see in the other species, and the mouse looks very much like this. So the consequence of this annotation is giving us a very clear understanding that way RNA, the average RNA molecule that's being made in the genome, is very much changing since the very earliest days when we had basically predictions of the RNA present in the genome, and highlights the fact that unlike the fly and the worm, the humans have very distinctive characteristics that we don't find in these genomes that have smaller lengths and number of CDS regions. So, in summary, the number of genes has increased from 37,000 to about 58,000. The number of transcripts increased from about 82,000 to almost 200,000. And most of this increase is in the non-protein coding long RNAs, genes, and transcripts. Now, this is quite important because at the end of the day, the number that isn't changing very much, right, is the gene regions here. The protein coding elements in the gene regions. Remember, about 19,000 of these are protein coding. That number has been fairly stable, around 20,000 for several years now. But this number continues to go up at a fairly geometric rate. And that number every year is almost increasing by 10 or 20%. So you would think that we had reached some asymptote, right, in terms of their saturation of things that we found. But in fact, the opposite is true. The more we dig into very specific cell types, the more we dig into cell work, and the more we have greater sensitivity of the RNA being made, the more we see various kinds of transcripts that are present. So by no means have we reached a sort of asymptote for the number of RNA transcripts being made by the human mouse genome. Now, one of the points that you could raise, and I think maybe in your own mind you have raised it already, is that what is this sort of these RNAs that are being identified? Are these new RNAs plentiful in the cell? Do we see many copies of these RNAs? All right? Or are they at very low levels? Well, this graph, which we published a couple of years ago, basically breaks up the RNA transcripts, the binding sites for transcription factors and DNA-1 hypersensitivity sites and other kinds of motifs, the enhancer and promoter regions, the transcription of chromatin marks and repressive chromatin marks. And if we plot the percent of the genome covered by each of these different kinds of RNAs, for example, poly A plus and poly A minus, and then we plot the percent of each of these bar graphs according to their levels of expression, you can see that a significant percent are barely being detected at something less than 0.1 FPKMs. And then from about this point on, the poly A plus downwards, you're over the 1 FPKM in the cell. And interestingly, this collection of RNAs called poly A minus RNAs, which virtually is ignored in almost every transcriptional study that you look at. Every time you read a paper and it says, we prepared the RNA by doing poly A capture of the RNA, basically this whole complement of RNA is virtually lost. All right? There's no information about that RNA. And those RNAs are quite abundant in the cell, and they are highly specialized as to the regions in which they're made. For example, the Phantom group, Piro Carnici's group, noted several years ago that the antisense transcripts that you see, genes that are overlapping other genes on the opposite strand, are more likely to be poly A- than they are poly A+. And like in many of these situations, this is not an all or none. This is not 0 or 1 kinds of things. There are clearly antisense that are polyadenylated, but the predominant profile of antisense transcripts is in this collection. So if you are interested in characterizing a gene region that you're working on, right, and all you work on is poly A+ RNAs, right, you may be missing transcripts that are buried in that on the same strand as a coding transcript or on the opposite strand, right? And these RNAs are in this pool of poly A- RNAs. And you can see that, in fact, there's the percent expression of these can be quite high. And this coverage right now is about at 80% if you take both kinds of RNAs. And the predominant amounts of RNA that are present in those regions are somewhere around about half of them is somewhere over about 0.5 ppm. Now, if you look at the rest of these elements that are being plotted, they too suffer from the fact that most of the signal that gives rise to detecting them, right, come from very low amounts of RNA-seq or chip-seq data. So in almost every instance, only a select proportion of the elements that are being mapped are at very abundant levels. Most of these are actually present at fairly low copy number or number of regions or signaled in that region. So, yeah, go ahead.
I think this is very useful for this question for this poly-RNA. You can see that almost 80%, even 90% of gene that expression is less than 1. Less than 1. That is most of gene not expressed, or a little bit expressed. Then the question is that, is this gene just expressed is less amount of expression? It's because our detection, because we just always check some cell type of RNA. Maybe some other cell type can be expressed, or the time points. You know, some RNA exists there, some genome is there. They may be just expressed in the, for example, in the embryo or somewhere in the adult. My question is that this very less expression, this RNA, is because the expression, is this the function? It just need not to be expressed that much, or is because that when we check that, we just check the special cell type or certain points. Do you think that this very last expression is RNA? They do have very important function, or?
Yes. So the point you raised is probably true on both levels. Namely, that we don't have a universe of cell types that we're looking at. Right? And most of the work that you see and we will talk about this in some detail a little later this morning, okay? But most of the cell types that are being looked at are either cultured cells, right, mostly transformed cells, or they're tissues, a heterogeneous collection of tissues, and where you're looking at the average expression of the many different kinds of RNAs that are being made in the different cell types. So if there are 100 different cell types in the slice of tissue you're looking at, all right, then that 100 different cell types, all right, are what you're seeing as their overall profile is the most abundant ones in each of those cell types, right? So it's both. It's the fact that we don't have a large collection of cells in a very individual fashion being tested, and we have this issue. So that if you look at the distribution of RNAs within a cell type, this is the HOXD10 region, and this is JAS1F. This is a very interesting gene. I won't go into it, but it's present at about 10 to 20 copies per cell. Its expression is at that level. This is work done by Jim Rock at the University of Pennsylvania, who has this very nice technique, has had it for many years, of hybridizing roughly 50 oligonucleotides targeted to different areas in a given RNA molecule. So each point of light here you see is a single RNA molecule with many, many different labeled probes hybridized to it. And you can see that at JAS1F, this mRNA, this protein coding RNA, is primarily seen as a cytosolic RNA in that locus. And what you see here is another example, and this brings home the point that you were making. Namely, in this population, if we were to take this population of cells that are expressing the HOXD10 gene and put a set of probes to the antisense of that gene, so look at whether there's a probe, an RNA being made off the antisense of that gene, you would see virtually no real expression in that population of cells, except in a very individual situation. In that very individual situation, we see expression levels that look very much like the JAS1F expression. But in this case, it is a nuclear transcript, and as you well know, and we'll talk about in a second, most of a large proportion of non-coding RNAs that we find in the cell are nuclear-oriented. So here is an example where if you have an expression of 1 in 1,000 cells, if the FPKM value comes out to be 0.001 or 0.01 rather, then you see that this is fleetingly present, all right, for that cell population. But in a certain cells, that program of expression is much different and much different regulated. The question then becomes, is this cell an aberrant situation, or is this cell providing information that the rest of its partners are not providing? And we just don't know the answer to that. That's the story. And as many of you begin to do work on single cell analysis, all right, you're going to see examples of this very frequently, where a gene in one cell, let's say you are surveying 100 cells in a given population, the same cell type, you think, all right, and all of a sudden one of these cells has a fairly high expression of one gene, one area in the genome. Is that an aberrant case for that RNA? In other words, there's some kind of misregulation going on? Or is that actually providing new information for the rest of the population that are around? And tomorrow, when I talk about RNAs that are exported out of the cell, I will try to make a case that this, in fact, is a common feature that we see where information is coming from minor members of a very large population talking to other cells. So, but we'll get back to that. So yes, your point is well taken. We don't have a universal coverage of RNAs, and even when we see low copy number, it doesn't mean that all the cells have virtually nothing in them, like one every so number of cells. In fact, some can have very many and look like a protein coding transcript. Okay, any other questions on this? This is actually quite important because many of you, I'm sure, in reading the literature, and somebody wants to make a point about a particular gene's expression, worry that the fact that it's low-level expression, why should we worry about it? We have enough to worry about things that are highly expressed, right? So why should we worry about low-expressing things? And so this idea of thinking about them being specialized, coming from very specialized cells, I think is an important thing to consider. This, the expression levels where we see this very low expression level can be sort of summarized in by dividing the RNA categories, again, up into coding and non-coding. If we take, again, just poly plus RNA, now these RNAs are, again, exemplified only of a very restricted percent of the RNA that's present from this ES cell line. And we look at the expression of the protein coding transcripts of RNAs that are present at lower levels, all right, but that have been validated independently. That is to say, this is not noise. These lower levels of expression is not a noise in the RNA-seq experiment that you're doing, all right? But in fact, by different other methods, by PacBio or by RT-PCR, you can see that these RNAs are present at these low levels, and they are clearly not noise in the system. And then these are novel, long non-coding RNAs that many of the RNA-seq experiments detect. And again, their average expression is at quite low levels, less than 1 copy per cell. This line here represents at 1 copy per cell. So they're down at something 1 every 100 cells or 1 every 1,000 cells here. This is kind of important because this kind of RNA that we see present in these cells, either have function or not. And is that function dependent on them moving their expression levels up, or do they operate at this very low expression? And we'll get to that point in a second. What's more striking to me is this plot that we published almost 4 years ago now at the end of ENCODE 2. And this plot is one of, I love this plot because it took me a long time to figure out how to do it. And what it is that it's a distribution. It's like this other diagram we just had shown, but it also takes into account where these low copy number RNAs are located. So if we look at the elements that are protein coding in the cell, right, you see that this distribution of expression is primarily central. Its centroid is in the cytosol. So most of those RNAs for protein coding are most likely going to be found within the cytosol of the cell. And its range of expression, you can see, is almost 4 logs, right? Across every point in that distribution, that grid distribution, is in fact present, can be present in either at something at 100-fold, or I'm sorry, 10,000-fold less than in other cell types. And these individual dots are individual cell types where you see the expression of this RNA present. Then you see this collection of non-coding transcripts, annotated non-coding transcripts, things that we have fairly good evidence are being expressed and are present in very specific cell types, all right, or in many cell types. And you see that the centroid of this distribution is much more nuclear than in the protein coding. And again, the average level of expression is now below 1 copy per cell on average for this distribution of RNAs. And finally, these are RNAs that have been identified fairly recently. So many of these RNAs are clearly found only in the nucleus, where the centroid is located. And the difference in its level of expression is almost 6 logs compared to the protein, the elevated levels of protein coding, which means that these genes have a variation level that allows for much more variance than the protein coding transcripts, right? And so we'll talk a little bit about what that means in a minute. But you can see here that you have cell localization information as well as the type of RNA being made and the place where that RNA resides. All right. So in summary, what we can say is that the characteristics of long and short non-coding RNAs are the following. The RNAs are arbitrarily defined as long and short based on a simple technique that can effectively separate RNAs that are 200 nucleotides from things that are less than 200 nucleotides. And in the eukaryotic cell, the mass of long-coding RNAs is greater than the short RNAs. Because 95% of the long-coding RNAs are ribosomal RNAs. But the molar amounts of short RNAs is much greater than that of long RNAs. So while the mass of short RNAs, you see in things less than 200 nucleotide, is much less than that of the longer RNAs, the number of molecules of these short RNAs, right, is many, many times greater than what we see of the long RNAs. And that's an important concept which we'll talk about tomorrow. But it will also talk about tomorrow is this difference between long and short RNAs. All right. Everything we've discussed so far are RNAs that are longer than 200 nucleotides, usually longer than 300 or 400 nucleotides. All right. That is only part of the picture. This point here, this comparison of long and short RNAs, it tells you that there's a population of RNAs that are either being processed from the long RNAs or being made specifically less than 200 nucleotides. And if you count the number of those molecules, right, in the cell, they are vastly outnumber what we see in long RNAs. And so I won't talk much about short RNAs in this lecture, but tomorrow I will talk more about it. Many the subtypes of long and short RNAs have preferred cellular compartments. For example, tRNAs are highly cytosolically localized, and long RNAs are in large measure nuclear organized. Many of the short RNAs possess a large range of modified nucleotides that are absent or less prominent in long RNAs. This is another feature that's not often considered, that the small RNAs are the targets for putting modifications on the nucleotides. In long RNAs, there are modified bases along the length of the RNAs, but they're very rare and they're very few. The most striking modification you see in the RNA molecules of long RNAs is in the caps, if the RNA is capped. We published, and a couple other people have published papers, that the 5' 7-methyl G cap modification is just the tip of the iceberg. There are many elaborate modifications at the 5' end cap structures of long RNAs. We have not a clue what those modifications are doing. Why? And they're very abundant. So the idea that there's only one kind of modification that sits in the 5' end of RNAs that are, when the synthesis of the RNA is complete, that is one member of a large family of chemical modifications that get put on there. And those modifications, for the most part, this is not certain because we don't really understand the pathways by why these other modifications are there and what they are. Those modifications frequently are put on in the cytoplasm, even if the RNA is intended to work through the nucleus. So you have this import-export system for RNAs specifically to get modifications put on them and then returned to the nucleus. Right? Yes?
I will give some comments in Chinese. I just want to go to the student.
Sure. Okay.
这个其实汤姆他其实是这个就是做我们这个 RNA 里边，你为什么说他在 RNA 里边我们的encode的计划，所有里边的 RNA 东西都是他很多的方法技术都是他做的，对吧？所以这块他做的会非常细，所以他今天的图给我一个非常大的印象。对 RNA 研究，我觉得你们看到他的图这张方向走了3个方向，我们很多东西都是错的，比如说朗姆特 RNA， 我们现在朗姆特 RNA 研究，我们觉得像最近这题大家都在做。最近疯子都在做到什么？做什么？所以你其实看朗姆特能控的人主要是在包核里边，你看到没？所以朗姆特研究主要应该研究它在核内的功能，而不是朗姆特研究我们其他的东西。当然我们可以研究，所以朗姆特我们大家不知道从哪下手，因为从它在核里边的作用，它主要在核里边，你可以知道最主要的功能应该在核里边发挥。所以朗姆特研究应该往这个方向走，这是一个。现在原来大家研究小的 R， 对吧 ？SR 小 R。这2年小的 R 基本没有，你可以看到自然基因指数基本没有，但是很少。但是今天他告诉你一个非常重要的现象是，小的 R 非常重要。它虽然说这样讲，它的这个分子的多少非常重要，而且小的 R 也是在核里，而小的 R 在核内的作用，就是我做的埋块，核内的作用可能是未来非常重要的方向。不仅是小的埋块，还有很多其他的，就是小的浓厚的 R， 不仅仅是长的，它的作用可能是非常重要。第3个修饰，杨云贵老师做了很多，现在这个 RNA 修饰那么多种修饰，他今天在后面告诉你修饰有多么重要，主要还在小的 RNA。你想他为什么要修饰，对吧？他修饰就是非常重要的，而这种修饰应该研究这种修饰的小的 RNA 和没有修饰的小 RNA， 它们什么作用？如果我们都 focus 长的 RNA 修饰，你可以看到它长的 RNA 很少，对吧？ 所以这就说明这个 RNA 研究，我们还是需要大师去指导，因为这个就告诉你很多的方向。我不知道你们听出没听出这一个东西来，而不是光获得知识。我觉得你要听课的时候要用脑子去想，它这里边跟我们以前接触的东西一样不一样，对吧？这个非常重要。I just tell students that more important that for non-nuclear function of non-nuclear, we should put more attention on the nucleus because this non-nuclear is most in the nucleus. So the function should be have that, but not some other. Another thing that, but now in China, a lot of RNA research, they put more attention on the non-nuclear RNA. But you can say that small RNA in the nucleus, the molar account is much amount. We should put more attention on this small nuclear RNA, including microRNA or some other nuclear RNA. Should be very important in the nucleus. They should have fun because they stay there. And the third, I told students that the RNA modification, nowadays a lot of modification, a lot of people have done that. The thing that this long multiplication is most in this small RNA. That means this small RNA, they have multiplication or no multiplication. The function should be different. So we should put more attention on this. So you also that maybe told us the three directions. We should do the RNA research. First, put more attention on the non-nuclear in the nucleus function. Put more attention on the small RNA. Put more attention on the small RNA multiplication in the function.
I was going to say that to you in Chinese. Okay, last point. Most short RNAs are derived from long RNA precursors, and the same short RNA sequences occur in multiple RNAs with different functional roles. So this is an important feature which we'll talk about tomorrow, this idea of creating small RNAs from long RNAs, all right? And what the fate of the long RNA is and what the fate of the small RNAs. And in short, I don't know in China if you're familiar with this, but there was a saying in America that no child should be left behind in terms of being educated and given, health care and things like that. Well, the cell has sort of a similar policy. No RNA, all right, is ever left behind. If you think about RNA as an entity that has many lives, all right, not just the life when it's synthesized, but lives after it's been made as a long RNA molecule and then created into small RNA is another life for that RNA. So we'll talk about that tomorrow also. Okay, I've been talking for an hour, so maybe it's time for a break and we can go back and take this story up again. Okay? Discussing some of the characteristics of these non-coding RNAs, whether they're short and small, right? And what I'd like to start with in this next section is to talk about the different kinds, the different types of RNAs that we see that populate different kinds of cells. Here we see a table which is segregated by the length of the RNA, and you can see it starts with these small functional RNAs. Now, the important characteristic of this table is that these RNAs are known to be operational. They are known to have a function inside of the cell. Then, currently, there's about 28,000 annotated microRNAs, and that they're primarily cytosolic in their subcellular location. And as everybody in this room knows, they're currently seen as regulators of transcription and translation. Now, we have to be careful about this because once we put things down on paper in tables like this, they sort of take on a life of their own. This is not to say that this is the only function, for example, of microRNAs. Right? There are other proposed purposes or functions for microRNAs, but they have less support than this. So my only objective in mentioning this is that when we characterize these things as functional and we give them a certain kind of function, please leave open in your own mind that these RNAs operationally can carry out other functions in addition to this. PiRNAs, many of you are familiar with this small class of RNAs. Again, just slightly larger about 26,000 piRNA regions are seen, and the nucleus is the center of operation to carry out transposon repression. tRNAs. We'll talk a lot about tRNAs tomorrow, but many of you have been, like I have, in their sort of earliest textbooks, molecular biology textbooks, see this as the small RNA molecule that's necessary for the translation of mRNAs into proteins. But more recently, these tRNAs are a golden example of RNAs that have multiple lives, and we'll talk about what that means tomorrow. There are about 600 annotated tRNA genes in the human genome. Snow RNAs, small nucleolar RNAs, there are quite a few of them, but there are about almost 500 nucleotides in length, the median length, about 750 gene loci. They are also nuclear in their localization, and they're responsible for the processing and modification of ribosomal RNAs and perhaps even mRNAs. There's some evidence for the use of these snow RNAs for processing of mRNAs. Then there's this class of RNAs, which my lab is very interested in, and it's called, as a group, the miscellaneous RNAs. And they consist of the yRNAs, the SCA RNAs, and the Volt RNAs. And they run up to about 100 nucleotides in length. There are about 27 Volt RNAs, and there are 4 yRNAs in the human genome. And they are clearly nuclear localized. And they are responsible for RNA modification, and yRNA has been implicated in, at least in vitro set of conditions, the ability to set up DNA replication sites in the mouse genome. And then long non-coding RNAs. There are, the median length is somewhere between the hundreds and tens of thousands. There are about 23,000, as we discussed. They are found both in the nucleus and cytosol, although predominantly in the nucleus. And they are clearly involved in structural regulatory elements. And there are about 18 well-established models for long RNAs that operate in that level. Okay, so after these different types that we talked about, in terms of the 23,000 long non-coding RNAs, the number of well-characterized long non-coding RNAs that we have a clear function for, all right, number is about is only about 120 of those 23,000. So clearly, there's a whole universe, all right, of discovery that needs to be applied to this class of RNAs. And every student in this room can look at this as an opportunity, right? An opportunity to think about what these RNAs are doing to explain some of the phenomena, some of the biological phenomena that you're looking at. Now, our goal here in looking for these functional properties for long non-coding RNAs is to basically, at the end of the day, define a role for the RNA. What is the role? What is the functional role for these RNAs? And therefore, what are the ways in which we can get to them? So this next section, is really a blueprint of how one might and has gone through steps in order to try to find a functional purpose for some of these non-coding RNAs. Okay, the first step is to build catalogs. And this is really what ENCODE is all about. You really want to know as many elements, non-coding RNA elements in the genome. And what does that mean? What it means in building a catalog is that you have precise initiation, transcriptional start sites, precise, if they're polyadenylated, where the polyadenylation sites are. We want precise splice points, which will give us precise isoforms. And all of that is critical in terms of looking for operating members that can be referred to if we're looking for a function that is explained by that RNA. So begin with the catalog of those RNAs. And this is, as I said, really the role of ENCODE, the ENCODE project. This project started, many of you may be aware or not aware, it started in 2002, about a year or so after the publication of the full-length working draft of the human genome. And there was a lot of hoopla about, goodness, we've now have, at that time, roughly 90% of the whole sequence. Now we have about 98%, 99% of the whole sequence. And that provided us a draft to do supposedly amazing things and have an effect on the health and welfare of human beings across the world. But it was clear that our ideas of where the operational units, the elements that carried out information, carried out regulation, was relatively unknown. I mean, many of you, perhaps in reading the literature, became aware of the fact that the first estimates of the total number of protein coding genes was 100,000. And people made a lot of money on that number, right? Because there were all these bets that said this number was wrong. Because this was an extrapolation of what we knew at the time. And so people's variation in that number ranged from more than 100,000 to things less than 10,000, right? And so it was uncertain, and it wasn't until roughly 5 years after the genome sequence was finished that the number of protein coding genes, their estimates, started to move toward the 20,000 number we see today. So this number has evolved in the last roughly 10 or 15 years to something that we have this assumption, or we have this working assumption that we understand where all the protein coding genes are. So I'm going to change this dynamic. Instead of me coming to you, I'm going to, if you're willing and I'm willing to participate, what is the biggest problem you think would turn that whole concept over? So, let's say we carry out these different kinds of assays, transcription, DNA hypersensitivity, chip experiments and all this. And we carry out these different experiments to look for functional elements, right? What do you think would turn this whole picture over? Give me one scenario which would basically change this whole idea, for example, that there are more than 20,000 coding transcripts, or there are more than roughly 100,000 enhancer regions in the genome. Give me one scenario that would change, a real scenario, one that the literature has, sometimes talks about, and there's arguments going on about. So today there is a way in which this whole scenario that we have here could be entirely turned over and change the dynamic of the range of things to look at. Anybody have a suggestion of what that would be? Is the question clear? I just want to make sure that the question is clear. Give me a scenario that would change these numbers that we have been talking about, the number of genes, protein coding and non-coding and so forth. Give me a scenario that would change those numbers dramatically. Remember, we said the estimates were 100,000 originally, now it's 20,000. What scenario would change that 20,000 number to something much different? What do you think? Come on, there must be somebody. Anybody? What do you think? Come on, think hard. All right, clue. All right. What happens, is there a threshold, for example, on the length of a protein coding region that we call it protein coding or not? Is there a filter that we put on that number? Anybody? Of course there is, right? I mean, if you see a transcript and it has the largest open reading frame of 10 10 amino acids. Most people don't think it's a coding transcript, right? Because we have this artificial concept that it would be a waste of time for the cell to make a very long transcript, right? And only get a 10 amino acid peptide out of it, right? So think about the fact that all these non-coding transcripts that we see in the genome, they have, some of them, quite reasonable length open reading frames, 50, 100 amino acids, right? But they either fail the length constraint or the codon usage constraint or the evolutionary relationship constraint, all right? And then we say, oh, it doesn't match any of them, so we don't think it's a protein coding element. What happens if the scenario is that the cell uses these RNAs for multiple purposes? So it has a function as a non-coding transcript, but it also has a function as a translated transcript, right? But maybe as a secondary function. That would turn this entire picture over. So as you're sitting there, probably bored to death with my presentation, you have an opportunity, perhaps in the near future or in the distant future, to see all of this discussion sound so silly. You'll be up here yourself someday talking to students and saying, gosh, I remember this guy got up there and told us there was 20,000 protein coding genes. Today we know there are 150,000, you know. And it's because the paradigm that we have for this is so built into us. We need to have sort of a golden standard that we hunger for this, and therefore we make this as a standard that if you want to change it, right, you're almost like a heretic, you know, in a religious sense, right? You clearly can't understand that the real truth is that we only have 20,000 genes, right? This is what happened when non-coding RNAs came up. People thought this was silly. If this was real, we would have known about this, we would have discovered it, and none of this would have ever, it's clearly noise, biological noise or technical noise. It's not useful at all. Don't even pay attention to it, right? We know there's 20,000 genes, and that's all we have to worry about, because splicing will take care of everything, right? But think about it. This is what we're talking about here. This is finding these elements is a door to finding if this 20,000 number is real or not, right? Because what happens if we expand this effort and do proteomics, and we start seeing proteins that don't match the coding regions, they match the non-coding regions, but they're only a little portion of that non-coding region. Think about what the effect would be if people had enough data for that to happen. The important thing of this conversation that I'm trying to get you to be part of is that you have to leave this open. You have to leave these concepts open in your mind. You don't have to act on them, but you have to not dismiss them. That's the important issue. Ideas need to be held, and they need to be validated, and they need to be understood at their implications, but they shouldn't be just immediately discarded, right? And no number, no principle, nothing I've said so far is unchangeable. Everything I've said is potentially changeable, right? So this is important to perhaps remember. All right. So back to biology. There are standardized steps to discriminate between protein and non-protein transcripts, and I just touched on some of them about the filters and the thresholds that are used to distinguish between protein coding and non. But in a sort of systematic way, we basically look for transcripts based on chromatin marks, based on 5'N sites, CADE sites, based on RNA-seq data, based on EST data, and based on other models that are mapping to the same region, we're seeing all this data. And from this information, we put together a set of transcripts, long non-coding RNA transcripts. And these transcripts can either be kept in the long non-coding category or put into the translational category. And that's clearly done by the length of the off. This is by non-random codon composition and also the evolutionary makeup of these RNAs in the off region. And in these regions that are present in the non-coding capacity, we basically come out to find whether there is a conservation of that locus, or is there a lack of conservation. Does it have any known, if it has a small open reading frame, does it have any known protein domains in it, and is it evolutionarily related to other known proteins? And then basically going down this branch structure, you decide whether an RNA is non-coding or not. This is pretty much what I just said, but the thing that perhaps keep in mind is that the evolutionary signatures of these RNAs turn out to be very important. One of the characteristics of long non-coding RNAs, beside the fact that you often find them in the nucleus, is that you find them in two very different classes. One is RNAs that are cell type and species type. That you only find it in a certain range of species of evolutionary time, and you only find it in certain cell types. Then the second class is one that you find throughout evolutionary phylum, and in fact in many different cell types. Right? So this concept of where non-coding RNAs lie, whether it's conserved or not, depends upon the RNA itself, what the RNA is. This is sort of underscored by the fact that if you look at the conservation of protein coding transcripts, right, they are obviously the most conserved. And the least conserved are the ancient repeat regions of the genome. And then the long non-coding RNAs are somewhere in between in terms of the amount of conservation that you see. So again, this reflects the fact that there are really two populations in there. One that have very long evolutionary history, and others which have a fairly recent one and very tissue type specific. This class of long non-coding RNAs that are cell type specific and localized, is easy to sort of see, but quite striking when you see it. The expression maps of long non-coding RNAs, for example, in this paper by Darren et al. For 31 tissues and cell lines, 10 of which are neutral. When you look at these different portions of the brain, which are categorized here on this slide, there's about 9 or 10 of them here. These different regions of the brain give rise to this expression pattern and this heat map. So each row here is a different gene, all right? And each column is a different tissue from the 31 tissues that were collected from the human patients. And this collection here that come from the brain have a pattern of expression in this heat map, which is very characteristic of the tissue that's present. If you closer look of these particular brain regions and the genes that are present, you can reorganize this into very area-related groupings. So for example, these two areas of the brain have relatively low expression level in this over the genes in this given region, and that is very unique compared to the other brain regions. So even within those categories, using just non-coding RNAs, you can see very specific cell type and region type specific expression. Now, this is another point that I'm going to stop the conversation for a second. this is another point. We're going to talk about this in a minute. When we talk about tissues, right, as being exemplars, all right, of that particular collection of cells that comprise that tissue, we're making a very large leap. Right? What we're doing is collecting a slice of the brain, in this case, right, which contains multiple cell types in that slice. And when you collect RNA from that slice of tissue, that RNA will contain RNA from each cell type, depending on what the number of a certain cell type in that population, all right, it's contributing a given overall proportion just on its cell number. But even if you were to think that each cell type in that slice was equal in their cell number, all right, all you'll see is the most representative, that is the highest expressed genes in each of the cell types. The lower ones will be diluted out. If you look at, if they're not equal number, and you have some cell types that are present at 1,000-fold less than other cell types, their expression, the highest expressing genes, will look very much like a very low expressed gene, right? But in that cell type is in fact a prominently expressed gene. So this is the caveats that we have to deal with on sort of a regular basis when the biological sample we're dealing with is heterogeneous, right, in its nature. There's one more very specific and very important caveat in this slide. Can anybody tell me what it is? It's kind of an important one, but very subtle. What do you think? Here's the person, right? Here are all these tissues from, obviously different people, all these tissues. What do we think is an important caveat in this overall? Anybody in this room want to give me their brain to work on? These are all coming from dead people. Do you think the pattern that's here is the pattern that you see in a live brain? Yeah.
Well, actually, I remember that there is some a small portion of functional neurons in the brain. The number of these neurons is very limited. So, for example, there's many neurons that have particular function. They have only one neuron for this specific function. So for this particular situation, we cannot get enough cell number to detect the non-coding RNAs or other RNAs specific for this functional neuron. So that will be a limitation for this research.
That's absolutely true. But in addition to that, to this very important observation, is the fact that this tissue, right, although in addition to being heterogeneous and either seeing just the prominent thing of every cell type or just a prominent profile of the most prominent cell type, is coming from an individual who died. Okay? That individual who died, all right, even if the ischemic time, the time from death to the time you collect the tissue and put it into some preservation state, right? Even if that's very short, is there any doctors in the room? Any MDs? Are there any MDs in the room? What is the ischemic? If somebody has oxygen starvation, like drowning or anoxia, right? What is the time to brain death in that individual? How many? Yeah, I think it's something in the order of, depending on the temperature of the individual. For example, people survive better if you drown in very icy water. Anyway, because your metabolism drops. Anyway, but I think in normal conditions, before you begin to see neuronal death in the brain, it's something in the order of at least 5 or 10 minutes. It's in that order. but maybe a little longer depending on the individual. But that's the realm before you begin to see real serious irreversible neuronal damage, or irreversible. So even if it were 10 minutes or 20 minutes, it doesn't happen like a switch. There's this cell response on the way to bad outcome, right? All of these tissues have very similar kinds of ischemic times until you reach, irreversible change in the tissue. Go look at the literature. Go look at the literature. Look how much of our understanding of human biology comes from autopsy material. It's virtually all of it. Right? And it's not a mistake. It's understandable, right? It's understandable. But it's something we have not really spent a lot of time thinking about or doing something about. And it's very important because in the end of the day, we could be greatly fooled by this kind of map. Now, the percent of genes that are unaffected, irrespective of the ischemic time, is probably not, depending on the length of the ischemic time, not tremendously large. But where you're likely to see the largest effect is in what? What class of RNA? What class of RNA are you likely to see the most dramatic effects? The ones that are a low copy number. The things that turn over rapidly. And which class is that? The non-coding class, the long non-coding RNAs. And in fact, there's now very clear evidence that in these autopsy materials, you see an underrepresentation of what is happening in the non-coding expression levels because of the turnover rate of those RNAs, and because these RNAs are very low copy number to begin with, so your chance of finding them when degradation begins to happen is even less. Okay? Yeah.
RNA standard, they use that people's brain to make the ERCC.
Yeah.
When you do the RNA-seq, you spiking.
Yeah.
So if, like you said, the ERCC's normalization or some quality control cannot be guaranteeing the expression of the non-coding RNA, right? Non-coding RNA, maybe we will mislead by this kind of reference standard.
Yeah, I think it's, it is one of those things when you watch people who are studying these kinds of things. There's always a control in there that seems to say, we looked at the rate of degradation, all right, of the expression pattern over if we leave the tissue unprocessed for longer periods of time. And we see this class of genes looks like they are more resistant to that. Therefore, they must be the ones that look like they look like this if it came from a healthy individual, right? Because they seem to be less resistant to the degradation pathways. But that's kind of, you in the audience can see the fallacy in this, right? Because if you unless you have a healthy brain to actually use as your standard, you don't know where the actual rate of you could have this very precipitous drop in the profile, all right, and then it slows down. So this rate of change need a positive control in which we know what it looks like in a normal individual, and then use that as a measure for the ischemic effect. Okay. Anyway, so I mention this again because it goes back to our discussion about leaving all things open. Right? When we say we have an expression pattern here that is very characteristic of the brain, we have to realize that this is a very specific biological state that we're looking at, right? And may not actually represent what a normal, healthy brain looks like.
So, I don't understand why do you think the shorter ones
are easier to get infected by death or life status?
When you say the shorter ones, What do you mean?
Because you just mentioned that the shorter RNAs are easily.
No, not the shorter RNAs. The ones that are that are relatively low abundance. Okay.
Yeah. But why only low abundance ones are?
Because
severe?
if you have let's say you have a fixed rate of degradation. Let's say one molecule of this class, all right, every minute. Just pick a number. Okay. If after 10 minutes you lose 10 molecules of something that has 100,000 copies, all right, you don't see very much of an effect. But if it only has 10 copies, you don't see it anymore.
But why do you think the loss rate is fixed rather than it's proportional to different?
Absolutely, it isn't fixed. That's the whole point. But for description purposes, I use this kind of example. It's not fixed. Each one will have a different rate, all right? And the different rate, right, its overall effect will be dependent upon where you start. But it's clearly going to go down, all right? So that's one. Okay.
Thank you.
Sure. So,
sorry, Catherine.
yeah.
I just want to add a comment here. Actually, most of the brain bank, they collect samples, and the post-mortem interview is actually more than 1 hour. It's a very long time.
Yeah.
And when we do the data analysis, we use the post-mortem interview as a control, just using the statistical method to control the fact. But we cannot make it as a normal, maybe like in an hour or less than 1 hour. This is the true situation in the brain study. And actually, the study in some groups, they just compare the people who died in their home, very peaceful, and compare the others maybe died by accident.
Mm-hmm.
So compare the microRNA is very different. And that's now the brain research is still using the died people. So
Yeah.
that's
So if I understand you correctly, what you're saying is that it's not only the fact that they die, it's whether they die at home or they die in an accident, you also have an effect on the effort. Yes. Yeah, and that's very true.
Yes.
That's so true. Okay. So basically we've been talking about this clustering of by tissue, right? And let's look by some very specific examples. For example, hot air, which is well known to you as one of the classic long non-coding RNAs, which is, by the way, an antisense transcript to the Hox region, one of the Hox regions, right? It itself is actually quite localized and very, very specific. Not only in the cell area, but where in an organism it's located. And so in humans, the levels of expression of hot air is actually highest in the lower extremities of a human than in the upper extremities of the human. And that's sort of a very gross level of localization. I mean, before, we were talking about whether it's in the nucleus versus the cytoplasm. But on top of that, you have the anatomy of the organism, all right? This also localization of long non-coding RNAs. Now, I'm going to leave this subject of clustering based on expression profiles of coding and non-coding transcripts, because I'm going to get to it, if we have time in the next hour, about that the use of identifying the origin of a tissue is based on its RNA is a sort of controversial and complicated question. So we'll get back to this in a second. I just wanted to make sure you understood. All right. So now we have this idea that we are doing classifications, all right? and we're building these classifications, and we're doing characterization, trying to characterize these long non-coding RNAs. But always we're moving toward the goal of finding out what is the biological function of these RNAs, right? And what mechanisms is used to carry out the biological functions, and do they have any relevance to human disease? This is, again, back to my original hypothesis that RNAs are really middle management elements, where very few of them, at very small numbers, in fact, control a lot of effects in the cell. Okay, so here's another example. Grace is a non-coding RNA. That is a gene that has a non-coding RNA that, as its antisense. This coding transcript encodes beta secretase, which is a key component to the synthesis of beta amyloid production in the brain. So if you have misregulation of this gene, you have increased production of beta amyloids in the brain. And that's been associated with Alzheimer's. And if you reduce antisense to Grace, what you lead is a reduction in the level of Grace in the cell. And what happens is that the antisense production of Grace is elevated in this disease state in the brains of individuals, allowing for this RNA to be elevated and to go down. So this is acting as an enhancer to the expression of Grace, and as you drop off the levels of the antisense expression, all right, the concomitant loss of the RNA is seen, of the coding RNA is seen. So right away, there is a lot of interest in building drugs that are targeting this antisense transcription. Right? But the key question is how? How do we do that? How do we develop therapeutics against an antisense transcript? And what are the issues associated with this? So is the question clear? So I'm asking you, you have a gene whose product isn't a protein. Its product is an RNA. And it's a clear regulator, all right, of a gene that is upregulated in the presence of Alzheimer's patients. All right? So if it looks like it could be a natural target, what is the issue associated with finding that as a drug target? What issues? There are two obvious ones. Yeah.
The first one is how you're gonna just specifically knock down the BACE1-AS. The second is the specific targets for the BACE1-AS. When you knock down this link RNA, it influence other mRNAs, other targets.
Right. So that's exactly the point. It's this issue of overlapping information, functional information in the same region. Remember, each strand is a separate unit when it comes to RNA, but they share the same information, so that if you mutate that strand, you mutate the coding strand, right? So you have the challenge of this overlapping structure of functional information that has to be dealt with. And why do we have to do it at that level? It's because the element that's functioning is the RNA. It doesn't become something else. It doesn't become a protein. It doesn't become a modification somewhere else. Right? So if you want to direct the therapy to it, you're directing it direct at that RNA level, which means you have to deal with the information that it's overlapping the coding transcript. Okay. Okay, so the the suggestion is that we could put some kind of variation in the promoter region of that locus, all right? But the promoter region of that locus, I'm not if you can see it. Yeah, here it is.
So the promoter region of the base 1 ES, not the original protein, but the antisense.
Yeah, this is the antisense. So this is exons 1 through 9 for the coding for race 1. And this is the antisense, codons 1, 2, 3, 4, 5. All right? Promoter is sitting here in the middle of this set of exons. So how are you going to change that without affecting these exons?
So put some DNA modifications on the intron part of the original.
Here?
Yeah.
Okay.
Or that.
That might work. That might work. But again, this splicing event is often conditional on what's going on in the intron too. So it might work, but then again, you wouldn't be surprised that it didn't. But go ahead.
Yeah, I presume if it's in other organisms, or other model organisms, we just develop a IAI antisense that much direct way.
But what are people who are, there are now billions of dollars, I'm not exaggerating, I said, there are now billions of dollars by established pharmaceutical companies and startup pharmaceutical companies looking at problems just like this, having to deal with it. What do you think they're doing? Let's say for the moment there's downregulation of this gene. Some mutation here causes downregulation of this antisense gene. What are you going to do? What can you do?
So try them in the mouse first, and then.
No, but let's for the moment not worry about where we do it first. Of course, we would do it in a model organism first, but what would you do? If you wanted to get this back up to a level that's normal, all right? What could you do without disturbing this protein coding transfer? CRISPR? What would you do? CRISPR-Cas9? You can edit the genome, right? You can get a copy of this gene, normal copy of this gene, all right, and put it over here if there's no annotated functional element there. Or put it somewhere where it might operate in trans. I don't know whether this only works in cis or can work in trans, so that's a very important consideration. But we now have the capability of editing our genomes, all right, to correct for things like this without and deal with this overlapping element. So you now put in a new element into the genome, right, to solve this problem. Right? Now, of course, you have to then put it into the germline, right? But you could also put it into ES cells, right? And then if the cell type that's affected, this neuronal cell type that's affected, could be driven from an ES cell, right? You could think of supplementing the activity that's lost through this mutation, through these engineered ES cells that would go down. That's the long-range vision. Whether we ever get there or not is anybody's guess. But it's up to you guys to actually figure out problems like this.
Since base 1 and base 1 AIs are both valid for AD and they are positively correlated, why not you target those both, but only you target the base 1 AS1? Thank you.
Yeah. So I'm not sure I understood entirely your comment. Could you say it one more time, please?
Okay, I said that since base 1 and the antisense of base 1 are both vaccines for AD, and they are the expression relationship is positive relation.
Yeah.
So why not target these two? You only ask question target one. I just think that they can target these two together.
Yeah, so I'm not an expert in this area, so I don't know whether changing this gene has any effect, all right, while you're trying to fix this one. So all we know is that this the expression of this is elevated, all right, in AD. All right.
Then maybe the side effect of changing base 1 AS1 is bigger than base 1. We don't know.
Yeah, we don't know that.
In this study, so the siRNA that is complementary to link RNA is also complementary to the gene of the BRCA1, right?
Right.
So how do you know that the reduction of the BRCA1 is not because of the siRNA also targets the gene of it?
Well, what the strategy that's actually depicted here is that if you have an siRNA control, right, both are expressed at relatively similar levels. If the siRNA is targeted towards the coding transcript, all right, you see this reduction in the coding transcript, and the anti-coding effect is unaffected by that siRNA. Right? If you take an antisense to the antisense, you now lose both the antisense and the sense. Right?
Yes, but that doesn't answer my question. So the antisense RNAs, siRNA, also can target the DNA of the BRCA1.
But the siRNA is the same sense as the sense. It is the same sequence that's in the protein coding. It's not. It isn't targeting the coding transcript.
Yeah, it targets the DNA. It can bind to the DNA of the BRCA1. So that may lead to the reduction of the expression level of BRCA1. That means that the siRNA of the antisense also can bind to the DNA of BRCA1.
Yeah, which it does.
Yeah, so which can maybe lead to the reduction of BRCA1. So if that's true, maybe the reduction of BRCA1 is not because of the reduction of the antisense link RNA, but because of the siRNA itself and deregulate the expression level of BRCA1.
No, but the siRNA does not bind to the RNA or the DNA encoded in the protein coding gene. It's the same one. It's the same strand.
Okay, all right. I will talk to you later.
Okay. All right. So another feature of this interesting long non-coding RNA is that it will actually repress other genes in trans. And so you see an example here where, excuse me, in the Hoxsey locus, the expression of these given gene regions, right, in the normal situation, right, is changed when you have alterations in the hot air expression levels. So all of these genes on chromosome 2 are affected, and you see the effect of the hot air locus in its expression when you alter its expression, and you see that expression is altered expression gives rise to this repression over here. And meanwhile, control regions are unaffected on chromosome 22. So this is an example of this kind of middle management where the same one element is actually affecting a whole variety of other different loci and the levels of expression. Yeah.
Also mentioned that this link is highly expressed in the lower part of the body, so the target of these other genes are highly expressed in the upper body.
Are you saying that they're still highly expressed?
I'm very interested in this link, the hot air. Yeah. And you said another slide so that hot air is highly expressed in the lower part of our body.
Yes, yes.
I think this is nearly impossible for me, but here I want to know the other genes, the targets of these genes, need to be expressed in the upper half of our body.
Yeah.
Is that true?
Yeah, I think this is actually all done in cell lines, if I remember the paper correctly, first of all.
Okay.
But second of all, although these genes are affected in this expression, in this particular experiment, it doesn't mean that in an in vivo situation, all of these genes need to be expressed. Right? What it means is that if any of them are expressed, they will be a focus of this particular antisense, of this non-coding protein.
But I think it is nearly impossible to be expressed only in the upper or lower part of our body.
Oh, I don't know about that. We can talk about this. So mechanistically, we see that the effect of this long non-coding RNA on genes that are in trans has a well-defined pathway in which that effect is achieved. With this hot air long non-coding RNA transcript is capable of doing is basically presumably through its structure, interact with RNAs, with proteins that are responsible for the methylation of protein in different portions of the nucleus. And it's also responsible for its interaction to Pol2 in its overall. This effect overall is due to the fact that mechanistically what this non-coding RNA is doing is acting as a scaffold for these modification proteins and to be brought to the nucleus and to the protein for modification. This is an entire new mechanistic paradigm which is only just beginning to be understood. If we look at hot air in terms of its effect in human health, we can see that if hot air is also present as an oncogene in breast cancer. If you have hot air present at relatively low levels, the effect is basically less dramatic in metastatic breast cancers than if the hot air expression is seen as at a relatively high levels. So its ability to act as repressors for different genes in the genome has a fairly dramatic outcome in terms of the progression of this particular kind of cancer. Again, showing that its overall importance, right, it far outstrips the fact that it's just a single gene and it has virtually no coding capacity. Okay? All right, why don't we have about 40 minutes left in class. Can we take a 5-minute break? We'll do it really short. Yeah, go ahead.
Thank you. My question is just like the slides mentioned, hot air is just a potential oncogene in breast cancer. My question is it possible in the future some drugs target to the hot air will combine to the drug like Herceptin to therapy in the breast cancer?
Yeah, so this interaction between Herceptin and the drug and its chromatin modification effects, this is something that people are very interested in. But what's really not clear is what the stoichiometry of this interaction is. How much of the drug is needed? How much does that effect have on the chromatin modifications? So this area is sort of like the area with the base 1 versus the antisense. We know some of the effects, but we don't know all of the effects. And I think this is sort of an entirely open area that says, here's a candidate, now we have to find a mechanism by which we can adjust the levels of expression of hot air in those conditions.
Thank you very much.
Sure. So we went through an example using hot air of how it is functional by interacting with these proteins and bringing these proteins to chromatin for altering the modifications present there. But in summary, we can say that long non-coding RNAs have a variety of known mechanistic operations. One is, as we just discussed, to actually, by virtue of presumably of its structure, right, interact with proteins. And then that interaction has some prominent downstream interaction within the genome or some other part of the operating cell. It's also been shown that these RNAs can interact directly with the DNA in the genome, and as well as with other RNAs. And EXIST is a great example of this, where EXIST was probably the first well-characterized non-coding RNA that wasn't a ribosomal RNA. Anyway, it codes the X chromosome, of course, and in doing so, it does it through both an interaction with proteins and interaction with the RNA and DNA levels of the genome. So I think this is a quite elegant example of each of these different kinds of interacting mechanisms. There are a variety of papers that cite the RNA interaction with multiple proteins, RNA's interaction with a single protein, and RNA interactions with DNA. And each of these papers have an immense number of controls that are needed to do this. So if you are involved in this, you know this very well, the number of controls that are necessary to actually prove this interaction is going on. And if you're not involved, then I would strongly recommend that you take a look at these and other papers, because it's not a simple set of experimental protocols. I want to finish really with one more set of interactions, namely the interaction in terms of regulation that non-coding RNAs have in the translational level. We think that RNAs, these non-coding RNAs, are primarily nuclear in their activity, but in fact there are a subset which operate at the level of the cytosol, and this RNA, UCH1, antisense RNA, this RNA promotes translation of a variety of RNAs based on a repeat SNV2 repeat, and this now is true. This SNV repeat element has been found in a couple of hundred now antisense transcripts, and are seen to be important in promoting the transcription of these downstream. And I think it's a very exciting paradigm that these non-coding RNAs are operating in. So there is a fairly open area for research which some of you are either involved in or might become interested in, namely ribosome profiling of long non-coding RNAs. And this came about by basically purifying RNAs that were bound to polysomes and to see what kinds of RNAs are present on the polysomes. Were there only the ribosomal RNAs and the messenger RNAs that are being translated? And so what the outcome was that basically that ribosome-associated long non-coding RNAs were quite numerous. There is a very large number of long non-coding RNAs that are pulled down when individual ribosomes and polysomes are pulled down. So almost 2,300 of them in this one experiment, where the levels of association that are either high or not associated with the ribosomes, indicating that there is a large repertoire of long non-coding RNAs that are somehow interacting at the translational machinery of the cell. And what they're doing there, whether they are very similar to what's happening with these symbiotic repeat containing RNAs or not. The promoting translation or regulating translation is still very much open and a very fertile area to work in. Okay, let's summarize this section, and then we can get into some of the things I said we would get to before it ends. Namely, the human genome contains thousands of non-coding RNAs, most of which have unknown function. As I said, only 126 out of the roughly 23,000 annotated non-coding RNAs have a very well-defined function. The non-coding RNAs are diverse in size, biogenesis, evolutionary conservation, and their function. And non-coding RNAs may help explain fundamental biological questions related to human evolution, disease, and genomic complexity. I think this is a wide area, and it starts in as an effort to indicate that these managers, these RNA managers, are looking for jobs, not so much, what it mostly means is that we're looking to identify what their jobs are, and we really don't know what that involves. Okay, so I have two more sections. We only have half an hour, and so if I don't finish these two sections, I will take them up tomorrow when we talk about circulating RNAs. Okay. Anyway, so let's talk about an area which we're quite interested in, namely this idea of character clustering RNA profile as to whether you can identify a certain cell type and its organ of origin. So we begin by basically saying that anyway, this is a recurrent challenge of the goal of precision medicine is to basically find very specific treatments for individuals. And you start with the patient, all right? You identify the organ that is in fact affected. You may be able to identify the particular tissue in this organ that is the cause of the disease. And you may in fact identify the specific cell type in the tissue that is the cause of the disease. And then look at the molecular level that's present. But the real question is, how accurate is the transcriptional profile of an organ or a tissue in order to define what normal is, diagnose the disease, monitor progression, evaluate drug efficacy, and define the prognosis. Does RNA profiling provide any or all of these kinds of information? And if so, at what level? Will the analysis of enriched constituent primary cells or a single cell be better? In other words, instead of taking the tissue, if we could in fact take individual cell types out of here and do a diagnosis on it, or take a tissue section and disaggregate those tissue sections down to individual cells, do we get a better outcome in being able to predict any of these diagnostic questions that we have? And what do transcriptional landscapes of constituent primary cells reveal? I mean, what do they, if ignoring the fact that we would like answers to these questions of prognosis and drug efficacy and diagnosis, ignoring those for the moment, if we look at individual cells, all right, or whether they be single cells or a collection of primary cells that constitute a diseased organ or tissue, right? Do we see something that reminds us of the tissue? In other words, can we deconstruct what we see with the tissue by looking at the constituent primary cells that make up that tissue, right? So the transcription profile is used to describe the phenotypic state of complex organisms. Right, about 50% of the 5,000 papers published in 2015 deal with this question. Basically, organs and tissues. We take a transcript from these organs and tissues, and then we take a population that's affected and a population that's not affected, and we say that we can identify the affected individuals versus the non-affected individuals by this differential expression pattern. This is about 50% of the 5,000 papers published in 2015 deal with this kind of approach. And in doing so, as we discussed earlier, the complexity of an organ, all right, is basically made up of a variety of different tissue types. So, for example, if we look at the lung, there are at least easily available through commercial means, at least 5 primary cell lines that are part of different sections of this organ that would be part of a tissue section taken from the lung. All right. If we take this transcriptional profile, disaggregate this lung into primary cells, and take the primary cells and do an RNA profiling on this, and ask, is this differential expression that we see for this any different than if we took the lung, or just the tissue itself, by looking at the primary tissues, right? And in doing this, our long-range goal, our long-range focus is that basically, considering the large number of cell types composing any tissue or organ, what constitutes the baseline normal, all right, transcriptional profile for the multi-component tissues or samples. In other words, what is a normal condition? All right, so what is a normal lung? All right, what is a non-diseased normal lung? All right, and do you have to have clinical phenotypes, right, in order to diagnose the disease, or is it already present in the phenotype of transcriptional profile? If we take single cells or low numbers of primary cells, right, will this allow for the identification of normal versus disease, and help more precisely identify the cell types involved in the affected condition? So in addition to being able to detect good from bad conditions, can we identify the causative cell type that's most affected in the disease state by doing this? All right? So we did an experiment, and we did an experiment with involving 53 primary cell lines. So these are homogeneous cell lines, all right, as defined by cell surface markers, collected from 10 different organs and tissues. The cells were screened for cell-specific biomarkers, the cell surface markers. There are 2 bioreplicas for every experiment, and ribodepleted stranded RNA-seq was used, and we had about almost 300 million mapped read pairs per replica pair done for these experiments. And you can see the number of samples here relative to the organ or tissue from which they were derived in the body location. Right. So the primary cells obtained from different anatomical locations is a real issue. When we ask the question, for example, we have 10 different primary cells in the lung, right? There are many different parts of the lung. And where that comparison of looking at what the profile for a tissue is versus what's in the constituent cell types really depends upon where the biological sample was collected. Was it collected up here close to the trachea? Was it in the bronchi? Was it in the small airway passages? You know, was it in the pulmonary riboblasts present in the lower extremities of the lung? Where was the tissue collected? And what proportion of these 10 different cell types is in that tissue? And this is true for all of the organs and tissues that we took a look at. So we tried in every one of these situations to pick the very same location in the 2 bioreplicas that we did. When we did the analysis by RNA-seq, we weren't surprised, but that individual primary cells have less complex transcription than the tissue section. So if you take a tissue section, right, and you do a profile on it, the number and the types of genes that are expressed in the tissue, right, is greater and more diverse than in the primary cells. That you would expect, and that's what we see. However, what we did see is that when you begin to cluster, right, the expression patterns, right, and you ask, does the clustering look more like where in the body location? In other words, if you had epithelial cells or fibroblasts or melanocytes, did you have them all come from a melanocyte would look different in the breast than it would in skin? Or did the embryological origin of the cell type actually contribute to its clustering? And what we see is that when you cluster the expression, right, the clustering that we see is almost entirely driven by the cell type, not by the origin or the body location. In other words, if you took epithelial cells, it wouldn't make any difference where they came from any one of these organs. It would look more like each other than if you looked at the tissue or anything else. So the clusters are not tissue specific, right? They're cell type specific, and that was a real finding for us. And you can see it here, where the mesenchymal stem cells, smooth muscle fibroblasts, all cluster together. The melanocytes cluster together. The endothelial cells cluster together, and the epithelial cells cluster together. Of course, all of these different organs in which they were taken from, all of these 10 different organs that were used. So they didn't cluster by organ; they clustered by the cell type that were present in each of these organs. And you can see this present in this reduced column that sort of segregates the clusters that are present. Strikingly, if you take another data type, in other words, DNase1 hypersensitivity, all right, in these different isolated cell types, they also clustered just like the RNA expression profiles according to the cell type, so that the open regions of the chromatin that are present mimic the expression patterns, right, of the cell type that were there. That dominates what's present compared to the tissue that was, or the organ from which it was taken. There were about 2,800 genes that are differentially expressed genes that are expressed in each cell type in each cluster. So that the driving forces for this clustering comes from about 2,800 genes. And if we look at that and look at the functional characterization of what those genes are, we see quite good confirmation that the genes that are seen to be developed, to be involved in epidermis development, keratinization, skin barrier, all are present in this epithelial cluster. Genes that are involved in the endothelial are circulatory and cardiovascular system and cell motility. The melanocytes are filled with genes having to do with pigmentation and melanocyte biosynthetic pathways. And finally, the fibroblasts are filled with muscle cells and collagen for an extracellular matrix at the center, as you would expect. So these driving forces are characteristic of the cell type, but they're not characteristic of the organ from which they came. If you look at the RNA biotypes, what you see is that most of them are being driven by protein coding transcripts, but there are about 300 non-coding transcripts which contribute to this overall cell type clustering that is going on. And in terms of percent, most of the RNA, non-coding RNAs make up less than roughly 10-15% of the defining gene expression. if you take the 2,800 genes and say which are the most important genes in that 2,800, it comes down to about 180 genes for which you can get the same clustering occurring. And what you see is that 180 genes, again, give us the mixed phenotype, the keratinocytes and epithelial tissues that are there. And if you do principal component analysis of the 180 genes, you can see that they cluster basically into 177 protein coding genes and 3 non-coding RNAs. If we use all 2,800 genes, you would have about 137 pseudogenes and about 325 long non-coding RNAs that basically give rise to this cell type clustering. And then finally, the endothelial-specific long non-coding RNAs, you can see the example of the non-coding RNA present in all these different organs, but only in the endothelial members that are present in each of these organs, whereas it's not present in any of the fibroblasts or epithelial cells or anything like that. And strikingly, if you cluster according to the transcription factors that are being expressed in these different cell types, you get back again these 4 clusters. These 4 clusters are seen as areas where you get transcription factor as well as DNase hypersensitivity similarities that are organized according to the cell type that are present. So again, very specific transcription factors are being expressed in very specific cell types. So what underlies all of this is the important feature that although organs, like a heart or a lung, if you do total gene analysis of those, you can see that this tissue came from the lung, or this tissue came from the kidney, or this tissue came from the heart, right? That turns out to be true because of the number of genes that are specifically expressed in that organ. If you look at individual cells, though, that compose that, what you don't see this picture of organ-specific clustering. What you see is cell-type specific clustering. So if you have a organ that's made up of 10 cell types, all right, and you take a tissue section from that organ, you don't get the tissue section profile by looking at the constituent primary cells. What you see is a profile that is indicative of the primary cells that compose the organ. And when you look at the whole organ, what you're seeing is this, again, filtered profile in which genes that are most abundant in the organ show up, and that therefore constitutes a picture of that organ. And that turns out to be quite important because at the end of the day, when you want to understand prognosis or you want to understand outcome for the disease or the presence of the disease, you're going to have some knowledge of the cell type that's affected. And you get that knowledge through these specific kinds of constituent primary cell experiments. So, in conclusion, the studied primary cells can be clustered into 4 major groups: endothelial, epithelial, melanocytes, and fibroblasts. The cell type clustering supersedes the effect of body location, the organ, or the embryological origin of the organ. There are 2,000 genes specific to each cell type cluster. Approximately 180 of these genes are enough to separate the cell type clusters into their individual groupings. DNase hypersensitive profiles mirror the gene expression clustering. About 25 to 50 transcription factors are distributed among each cluster, and they drive the organization of these clusters and mimic the gene expression. The correlation of primary cell transcription with the whole tissue profile is poor, and either there are missing primary cells, in other words, we don't have enough primary cells to see something that looks like the organ, or, and most likely, the reconstruction of tissue profiles be a complex integration of data from parts of the primary cell profiles. In other words, there's a distraction of information from the primary cells that we need to carry out in order to reproduce what we see in the organ level. And so this has very significant repercussions for how using transcriptional information to make diagnosis and prognosis activities. Does that make sense? And does anybody have any questions on that? Importantly, long non-coding RNAs look like they, for this particular purpose, look like they're not a dominant factor in most of these this situation. Any other questions?
So I don't understand why DNS
expression of the clustered genes. What's the underneath mechanism?
Well, I think it follows fairly straightforwardly. If there are very specific members of the, let's say, the 2,000 gene cluster, all right, that's divided up into, so portions of that 2,000 genes are specifically found in epithelial expression. Portions are found in endothelial expression. Those genes are active, and those genes will have loci, all right, that are indicative of expression, all right, and be available for activation. All right. So it follows that when you look at a epithelial cell, it will have the same genes and therefore the same hypersensitive regions, all right, as the expression pattern. So I think it follows from that directly.
But we know some instances that DHS do exist in those or near those not expressed or silenced genes.
Yes. Right.
This, I suppose, is a very small.
Right. But here, when we look at the expression, the DNase I hypersensitivity sites, they're dominated by the availability that is concomitant with the expression. So you would see the promoters of those, particularly, you would see some portions of the gene body being the dominant kind of DNase I hypersensitive region. And these others add signal, but they're not consistent in terms of clustering because of the expression pattern of the genes they're in. So I think they just go hand in hand together.
Thank you.
Yeah.
We discussed the brain tissues earlier, and there are many different cell types in brain, like the neuron and the glia and other cell types.
Yeah.
So what do you expect about the brain cell type expression in your data? I didn't see any brain neuron samples in your current analysis.
Right. So this study was basically driven by the cell types that we could collect. All right. So we got these cell types from commercial vendors, right? And I don't know of any vendors that provide neurological cell types. So there are people who are collecting these different cell types from the brain at individual levels and doing single cell kinds of experiments. And that's what we're working on now, is doing cell surface markers of brain cells, right? And then using those markers to get enough cells to do the profiles of individual brains. The question we would like to know is, as with these other organs, does a chandelier neuron, right, in the hypothalamus look like in the neocortex, right? Or do they actually look exactly like, irrespective of what portion of the brain? That's one of the questions we're trying to do now. Yeah.
So, for the
tissue-specific link RNA, does anyone look at whether the histone modifications and demethylations is also similarly regulated to the tissue-specific genes, tissue-specific mRNAs?
No, I don't think I've ever seen anything that basically, in other words, I don't know of any specific data that suggests that if you have a cell type or tissue-specific long non-coding RNA expression, do you have a pattern of chromatin modification that is also tissue-specific? And I don't know of any data that's been reported about that. Thank you.
For your last part of the story, you found 177 coding genes and only 3 non-coding genes.
Right.
So my question is, maybe you remove these 3, it will be better to classify and characterize the tissues. My second question is, I'm still interested in the nuclear located non-coding RNAs, and you saw some evolutionary signatures of the coding RNAs. And we want to know some evolutionary signatures of these non-coding RNAs. And thus, I'm more interested in the sensor of these RNAs. And can these sensors sense the RNAs and take them out of the nuclear and back to the nuclear? And what do you think? What do you think the factor that determine the subcellular location of these RNAs? Thank you.
Yeah. So you asked several questions in that, so I'm not sure I remember all, but these RNAs, their localization is clearly tied to the proteins that they're interacting with. So the proteins are operating in very specific areas of the cell, and part of that happens by virtue of the fact that the translation of that protein, all right, is also localized. This is very clear in the neurons, all right, where ribosomal polysomes are actually being transported to different portions of the neuronal cell, all right, and it's in those particular areas which very specific proteins that operate in those areas are located. And it would be, I think, quite reasonable to think that translation, right, is accompanied by a means of selecting from the RNA population that's there, given partners, right, the non-coding partners based on their structure, right, that allows them to migrate to even more defined areas in that given locus. So I think it's the interaction with proteins that help determine the localization. And the second question is, the sensors of how one is picked out and how, if that's your question, I think resides in the overall structure of the RNAs themselves. That seems to be the most likely case, but what is relatively unexplored, I think, is the fact that whether there's any concomitant modification of the RNAs that is also recognized in addition to the secondary structure or the tertiary structure of the RNAs. And that's relatively unknown at the moment, because the ability to isolate from in vivo sources enough RNA to determine what the modifications are present on specific molecules is a capability we don't have as yet. But that's an intriguing possibility because we say that because it's a sort of fallback to the idea that if it's the structure of the RNA, we don't know a lot about how to predict or model these structures very well, right? We can make hairpins, we can make hairpins out of anything, but really what's being recognized is a much more subtle, much more sophisticated kind of interaction, and which is dependent on the sequence. And I think that is still rudimentary in our understanding. Yeah.
Hello, Professor. I am very interested in the translational potential of the non-coding RNA defined by the previous pipelines. So in your slides, you discussed the polysome-seq just purify the RNA separated, co-purified by the polysomes, and do the chip assay or CP assay. I think these linked RNAs identified from this assay might be indirect because they just show the associations. So have you checked, or do you know somebody else do the RiboSeq? They just digest the to digest the ribosome and RNA complex to purify their footprints to map back to the linked RNAs. Maybe this approach where we can identify the direct sequence or the mapping reads of the linked RNAs. Maybe these linked RNAs they can have more possibility to translate peptides. Maybe these definitions may need to change.
So I know in the literature there are multiple papers which have done fairly sophisticated analyses of mass spec data from cells, looking for given peptides which are predicted to be possibly translated from long non-coding RNAs. And those papers have very clear evidence that some of these peptides, which can only appear in these sequences of the non-coding RNAs, seem to be in the cell. The other thing that's sort of persuasive is that the RiboSeq experiments that have been done, right, have also been done in a way in which they've done riboprotection experiments, where basically the ribosome, right, is treated with RNase, right, and so any RNA that's exposed to the RNase, all right, is removed, and then whatever RNA is left, all right, undamaged by the RNase, is then characterized as being a protected portion of some RNA, whether it be coding or non-coding. And what we see in these papers that report is that the protected parts seem to correspond to the sequences of the peptides that are being identified, indicating that somehow there is a correlation between what the ribosomes are doing with that RNA and what the product that could be coming from that. So there is a consistency in that story. It is hard, though, in some ways, because the abundance of these, again, it comes down to this question of abundance. The abundance of the long non-coding RNA and the abundance of the peptides are very, very fleetingly small. Right? So very deep sequencing has to be carried out, even though you have the enrichment of the loci, of the ribosome loci, helping you identify them. But it's also true that the mass spec data is relatively inconsistent in being able to see it. So the same sample done 3 times may only give you that peptide once, right? And with the possibility that there's not a lot made, so you don't see it all the time, right? So there is very much an open question of whether this is really indication of the coding potential of these non-coding RNAs and what they might be doing in associating with the ribosomes. Yes.
Hello, Professor. In your slides, and you show the relationship between RNA and some disease like AD and breast cancer. But I'm very curious because we know some genetic disease is related to the defection of genome, such as amplification or deletion. So my question is, have you got some data or some research on the relationship between RNA expression and genetic disease? Yes.
So the literature has several very good papers which clearly show that that when the GWAS, and you know what GWAS is?
I don't know. Please explain.
Okay. So GWAS is this study that went on for about 10 years now, where the genomes of many individuals, humans, have been sequenced to look for variation in the human genome. So for every base in the human genome, how often do we see a variation at that site? Is it 5%? Is it less than 1%? You know, so if you have 100 people, right, and for this one place you have an A, all right, but in one individual you have a T, all right, so that's a 1% variation rate for that base. Okay, so that study has been going on for 10 years. So what are the common variants and what are the rare variants in the human population? You know, and how does it segregate by the different ethnic groups in the world? Okay, so that collection of data clearly shows that the variation, most of the variation you see is not in the coding regions. All right, more than 90% and 95% are in the non-coding areas of the genome, and many of these areas are areas where non-coding RNAs are made, and also where regulatory elements like enhancers and promoters are found. And so these variations that are found have been related back to disease states, very different disease states. And so there is now a wealth of papers that clearly show a high correlation, a statistically meaningful correlation between variants that occur only with this disease population and not with people who are unaffected by a disease. And many of those variations occur either in a regulatory region, all right, or in a non-coding RNA region. And those that are in the regulatory regions are being studied for ways in which to recapture the effect of the variation. And then non-coding RNAs, the question is, can we provide ways in which that RNA, first of all, understand what the function of that RNA is in the disease, and then provide substitutions for that the varying RNA molecule. So there is a lot of information in the literature that addresses the question you mentioned.
Thank you.
Okay, so it's a little afternoon, and I hope this morning's meeting we had was useful and helpful. I want to repeat that if there are any questions that you think about when you come to go over this lecture, then please feel free to email me, and I'd be glad to answer back and answer any of your comments or questions. Thank you very much.
I would like to have a small briefing at the end, so Thomas. Actually, one thing I forgot to mention is when I introduced Tom is Tom spent the whole night to make the slides to look better and to fit the windows. So let's give a warm thanks to Tom again. 好了，那我们就去吃饭好了。如果有同学愿意还有问题的话，可以聊天。